So hi everyone. I'm James Salazar. I'm one of the third year cardiology fellows and the chief fellow that is helping lead cardiology grand rounds this year. Very excited to kick off this year with Mikhail Alexanian. Um, just a little bit about Mikhail, who has a great talk lined up for us today. He is an investigator at Gladstone Institutes and an assistant professor at UC San Francisco. Mikhail completed his training in Italy and Switzerland, receiving a doctorate in stem cell biology at the University of Lausanne in the lab of Thierry Petrosini. During his doctoral studies, he investigated the role of non-coding RNAs, including transcribed enhancers and long non-coding RNAs in pluripotency and early cell fate specification. He joined the Gladstone for his postdoctoral work, where he worked under the supervision of Deepak Srivastava and Saptarsi Haldar and discovered gene programs related to cellular plasticity in the context of heart failure. He now runs an independent lab that is interested in understanding the genetic and epigenetic mechanisms that govern cell identity in health and disease, with a particular focus on heart failure and dissecting the gene level changes that underlie heart failure, with the ultimate goal of developing targeted therapies to preserve or restore heart function. So with that, Dr. Alexanian, welcome, and we're very excited to hear your talk. Again, just to remind everyone, the talk will be about 45 minutes, and then we'll open it up to questions at the end. Feel free to drop questions in the chat as we go along, but we'll reserve it until the end. I'll hand it over to you, Mikhail.
Thanks, James, for the very nice introduction. And I'm very excited about the opportunity of telling you about what we're doing in the lab. And I thought I would start with this slide that really highlights the sort of the price of success of the increasingly good cardiovascular care. Of course, you know this much better than I do, that the increasingly good cardiovascular care, which probably makes your patient likely not dying of their first myocardial infarction, leave us with this huge population of people with chronic heart failure. This is really an epidemic, which has a huge burden in our society. Now, heart failure, as you well know, it's characterized by pathological remodeling and many pathological features. Some of them are intrinsically related to muscle cells in the heart, cardiomyocytes, which they become dysfunctional. In many instances, they go into hypertrophy. There is electrical remodeling. There's certainly a signature for metabolic dysfunction in heart failure, and a strong signature for inflammation and fibrosis. Now, where my lab comes and with the background of molecular and cellular biology is really trying to understand these pathological processes at a cellular level, with the ultimate goal to really try to identify molecular mechanisms that are critical in heart failure pathogenesis, with the idea of ideally find new druggable nodes for this disease. And I should maybe highlight that a lot of the research and in basic science related to heart failure, and generally I would say cardiovascular science, has focused on cardiomyocyte biology. We know a lot about the genes that are driving cardiomyocyte hypertrophy and dysfunction in heart failure. We know much less about the role of gene activation and pathological response in other cells in the heart, like immune cells and fibroblasts. And today I'll tell you a lot about our work related to inflammatory and fibrotic processes. Now, heart failure is notably associated with a lot of inflammation and fibrosis. And again, you're very familiar with that. Structure, this fibrotic response, And we know that inflammation and fibrosis are two processes that are extremely connected to each other. And this is true in heart disease, but in many other human disease as well. And generally, there's the notion that there's various pathological insults. They drive immune cell infiltration from the circulation to the heart. These immune cells, they promote an inflammatory processes where they will secrete factors like cytokines and chemokines. And we know this drives inflammation, but we also know that this is deeply intertwined with the fibrotic process. But I would argue that the molecular mechanisms that control how the inflammatory cues can promote fibrosis are really not well understood. And I'll go back to this in the second part of my talk. Now, instead, in the context of fibrosis, we know that these tissue processes is the result of the uncontrolled activation of a cell type called fibroblast. Fibroblasts are everywhere in our body. They have an essential role in maintaining the tissue structure, and they're also very important in acute injury response, for example, wound healing. Now, the issue with these cells is that they, in the context of chronic stress, they transition to an activated cell state that usually it's referred as activated fibroblast, or you might have heard myofibroblast in this context. And this cell type is really the one that promotes the fibrotic response, which we know being associated to cardiac dysfunction. So what are these activated fibroblasts doing in the context of heart failure? Well, they proliferate for sure. They start expressing proteins related to contractility, so they start to contract. This is really one of the pathological hallmark of the fibroblast response in heart failure pathogenesis. I'll show you some data on alpha-SMA, that it's one of this contractile protein that fibroblasts activate upon stress response. And then maybe the most known response that fibroblasts undergo in the context of stress is this excessive deposition of extracellular matrix proteins like collagens. And here I'm mentioning periostin because it's a protein that I will talk about in the following slides. Now, this is what happens histologically, and I guess you as physicians are very used to see histological assessment of either inflammation or fibrosis, but myself, as a molecular biologist, I think about this process as fundamentally driven by changes in gene expression and cell states. So the genes that usually are referred to promote fibroblast activation and fibrosis are called profibrotic. And here on the left, you have an image of a mouse heart that it's healthy, that has a very low fibrotic response. This is the picroserious red, is a way to assess fibrosis in histology. And these profibrotic genes are turned off, they're not active, while in the context of this strong profibrotic response in a model of murine heart failure, there is strong fibrosis and these profibrotic genes are activated. So given that I will talk about how genes are getting activated and suppressed in the context of heart failure pathogenesis, let me take just a minute to remind you a few core principles of molecular biology, where we know that all the cells in our body, they share the same DNA. What makes the difference between, for example, a bone cells and a nerve cells is the set of genes that are activated, that are on in these two different cell types. Now, I think that we're used to think about cellular plasticity as a natural feature of cells in cellular differentiation and embryogenesis and development. So, for example, you have stem cells here on your left. This is a cell that can become any other cell type with the right cues. And the stem cells, if receive the cues to become a neuron, will activate a set of genes that will promote this cell state transitions into a neuron as well. It could activate gene programs related to skeletal muscle or cardiomyocytes. Now, you have to think about that even our adult tissues, they retain a certain level of tissue plasticity. So you can think about that pathological stress can indeed drive changes in how the cells function and in their shape, what I will refer as cell state transition. And here below, I'm showing again as an example the fibroblast. On the left, a healthy fibroblast. And through the stress programs that are activating these cells and the pathological genes that are activated, this cell transition to a disease cell state, which is the activated fibroblast. And if one would ask me to really, have a very, easy summary of what my lab does, it would be the following, the one I'm showing you in this slide. We're really studying what are the molecular mechanisms that drive the expression of those pathological genes that can make transition healthy cells to disease cell and how this is associated to organ dysfunction and disease, with a final goal of identifying those regulatory nodes that are important for this transition, with the aim, of course, of exploring these nodes as possible therapeutic approaches. Can we block, for example, these regulators to stop this cell state transition or even revert it once it has already happened? Now, in the last few years, we became very interested in a particular protein called BRD4, which in the context of this pathological cell state transition really has an important role. And this protein is called BRD4. The work that I'm going to present around this protein has been led by myself and someone you know very well in your division, Arun. And here I'm showing a few of the manuscripts that we've published together, which are all centered around the theme of studying pathological gene expression in the context of heart failure pathogenesis. Now, what is BRD4? Let me tell you what this protein does in a very easy way. So BRD4 is a protein that it's able to activate genes to promote their transcription. And what we know about BRD4 is that it binds the DNA and activates the transcription of pathological genes, so those genes that drive remodeling in heart failure. And we have exquisite modalities to actually block BRD4 function in vivo with a small molecule called JQ1. So from now on, when I say JQ1, you have to think about this molecule that it's able to inhibit BRD4 function. So what does that mean? JQ1 will bind to the pocket that BRD4 protein uses to bind these genes. And as a consequence, it will displace BRD4 away from these genes, will kick it out. And these pathological genes will be suppressed. So we'll go from an on to an off state. And work in the last decade for our group and many other groups have really proven that the inhibition of BRD4 function with JQ1, it's protective across several murine models of heart failure. And here, I'm showing you an experiment from a paper in 2017. This is the use of JQ1 with this BRD4 inhibitor in a mouse model of pressure overload called TAC, which stands for transverse aortic constriction. I will refer to this mouse model in the next few slides. The way it works is that our mouse cardiac surgeon applies a constriction at the level of the aorta, which over time will generate a pressure overload. And this model, over time, it's associated to systolic dysfunction, to increased fibrosis, increased inflammation, and cardiomyocyte hypertrophy. And you can see this huge hypertrophic response here on the left in the vehicle group from sham, which is the control. There is no TAC surgery here. And TAC, you can see the strong hypertrophic response. And I hope you can appreciate how this BRD4 inhibitor, JQ1, really suppresses this hypertrophic growth that it's driven by TAC. And JQ1 is able also to suppress in these experiments inflammation, fibrosis. And so this this really highlights it, how we can modulate, we can turn off pathological genes in the cardiac tissue to achieve amelioration of cardiac function. So the natural question would be, well, why don't we use GE1? Why don't we give GE1 to patients? Well, the issue with GE1 comes from the fact that BRD4, it's a pleiotropically expressed protein. It's expressed pretty much in every cells of your body and has important function also. So the use of GE1, it's usually associated with a strong on-target toxicity on off-target tissue. And that's really why we can't, for now, use a molecule like GE1 for treat heart failure. But what we can do is to use it in the context of basic research to understand what are the downstream targets of this important regulator BRD4, with the hope of finding regulators of pathological gene programs that might be more cell type specific and likely more amenable to therapeutic intervention. And this was really the goal that we set to explore a few years ago with this project that we ended up publishing in 2021. This is the first experiment we're done. Again, this is the model of pressure overload murine heart failure by TAC. In this graph on the y-axis, you have the left ventricle ejection fraction. In the x-axis, the time. Those are days after the surgery. And you can see in green, the sham control, no heart failure, and in red, the TAC. And you can see there's a classical decrease of EF over time. So what we did in this experiment was that we took a group of mice that undergone the TAC surgery. And after 18 days, where you can see the cardiac dysfunction is already established and the function is already down, we started administering this compound GE1. And you can see what happens here in blue. It's a pretty striking response. The EF basically goes back to the control. Now, the half-life of GE1, it's very short, meaning we have to inject GE1 every day in the mice. We inject it with an intraperitoneal injection. And in the context of this experiment, we wonder how dynamic the effect of this BRD4 inhibitor was. So we took a group of TAC GE1 treated mice, and in the last 2 weeks, from day 45 to day 60, we just stopped treating the mice with GE1. And that's what happens. The EF rapidly goes down in the orange group, highlighting almost how we can toggle the functions up and down by starting and stopping the treatment with the BRD4 inhibitor, GE1. And this has been very puzzling to us. How does it work? What are the cell types? What are the cell states that are the target of BRD4 inhibition? And even more importantly, can we discover novel biology? Can we discover new genes and molecular mechanisms associated to heart failure pathogenesis? And to address this question, we used a technique that I would say has been one of the biggest revolutions in the last decade in molecular biology and cellular biology, single-cell RNA-seq, that allow us to measure how much a gene is expressed in every individual cell. And as I will tell you about single-cell RNA-seq and show you some results, I want to take a minute to tell you what it is and how you should interpret a single-cell RNA-seq figure. So what we do is that we take the cardiac tissue, we take the heart, so we can do that in a mouse, but we can do it also in a piece of human heart, and we dissociate this tissue into single cell, and then we run this single cell sequencing. And that's usually what you would see in a single cell RNA-seq result. It's a figure that looks like that. Now, in this figure, every dot is a single cell. And the closer the cells are to each other, the more similar the genes that are activated will be. So two dots that are very close will express similar genes. Dots that are very far apart in this two-dimensional space will instead express very different genes. So here I'm showing you an example of the cells in the heart that we can find. There's a lot of vascular cells. There's, of course, cardiomyocyte, immune cells, and fibroblasts. And of course, they're all separate because they're different cell types, so they express different genes. So what we've seen in the context of our single-cell RNA-seq experiment in the heart failure model TAC, with and without GE1, was pretty striking and in a way shocked us. The cardiomyocyte, which are as I mentioned before, the cells that have been probably the most studied in the context of heart failure didn't have a lot of changes, a lot of genes changed associated to the use of GQ1, suggesting that most of the direct function of the BRD4 inhibition might be happening in other cell types. And the most striking changes were happening indeed in the fibroblast population and in the immune cell population. And I'll talk about these two cell types from now on. So this is how a single cell RNA-seq result will look like. These are fibroblasts. Again, every dot is a different fibroblast. And again, the closer they are, the more similar they are in the genes that they're activating. And in this context, the fibroblasts have been colored by sample identity, meaning that the fibroblasts from the healthy heart, from the sham control, are in green here on the left. The fibroblasts from the TAC heart failure are in red here on the right. The fibroblasts from the GQ1 treated group are in blue here below on the left. And the fibroblasts from the TAC GQ1 withdrawn group are in orange, and you can see where they are distributed. And I draw some arrows to highlight the different transition that the fibroblasts are undergoing in this context. So you see that in the context of our failure, fibroblasts move from this healthy state to these red cells here that are clustered very separate from the sham fibroblasts, which are in green. When we use the GQ1 in the continuous mode, the fibroblasts almost go back closer to sham, meaning that the genes that are expressing are, now different from TAC, and they're getting closer to a healthy fibroblast. And when we stop the treatment with GQ1, and if you remember, the function goes down, we see this fibroblast moving back, or at least most of them in orange, towards the TAC signature, towards the signature in red on the right. So let me show you what that means in the context of the expression of a specific gene called periostin. Now, again, here on the left, that's the same figure I showed you before. Those are the fibroblasts from this experiment colored by sample identity. And that's the expression of this gene periostin. Why am I showing you the gene periostin? Periostin is considered the best marker of fibroblast activation in heart failure, meaning this gene, it's very poorly expressed in a heart that doesn't have fibrosis and hugely activated in a heart that has a lot of fibrosis. And here you can see the expression pattern of periostin in the same fibroblast that I'm showing you here on the left. And that's why the shape is similar. Now here there is a scale where you see orange, it means that periostin is strongly expressed. Where you see blue, it means that it's lowly expressed. And you can see how most of the expression of periostin comes from the right side of this two-dimensional space, exactly where most of the red and orange cells are coming, right? The cells, the fibroblasts from the TAC and the TAC GQ1 withdrawn mice, which are the one with the systolic dysfunction. And here on the right, there's the quantification of periostin expression. This is called a violin plot because this figure, they look like violins. And you can see in the y-axis, there's the expression level of periostin, meaning the more dots you see and the higher these dots are in the y-axis, the more periostin is expressed. And you can see two violin plots for each condition because we performed this single cell RNA-seq in 2 separate animals per condition. So a total of 8. And you can really see how indeed periostin is strongly activated from sham to the heart failure model TAC, strongly suppressed by the BRD4 inhibitor GQ1. And once we stop injecting GQ1, periostin is upregulated again. And periostin, which I'm showing you as an example, behaves exactly like hundreds of those profibrotic genes that are known to drive tissue fibrosis. They all behave in the same way. So what this data suggests that by inhibiting BRD4, we get not only block this transition from a quiescent fibroblast to an activated fibroblast, but even revert it once it has already happened. And this has, of course, effects in tissue fibrosis, which we have measured in the context of this same experiment, showing that indeed GQ1 decreases tissue fibrosis by histological assessment, while the withdrawal of GQ1 is associated with re-increase of tissue fibrosis. So where do we go from here? So we use this different data set to ask the following question: can we find a novel regulator of fibroblast activation in heart failure using this data set? And to make a very long story short, and if you're, of course, interested in the details, you can go back to the original work in more detail. What we found, it's a gene called MIOX1, which was completely unknown in the context of cardiovascular science and heart failure, and generally really poorly known in human disease. This gene is called MIOX1, and here I'm showing you on the left the same fibroblast figure that I showed you before. So these are the fibroblasts that are colored by sample identity, and that's the expression of MIOX1. So if you now have in mind the expression pattern of periostin, you'll recognize that the expression of MIOX1 really overlaps with periostin, meaning that MIOX1 is specifically activated in heart failure in activated fibroblasts. And you can see the quantification of MIOX1 expression in the Violin plot, where you can see that in sham hearts, the fibroblasts express very low level of MIOX1. MIOX1 is strongly upregulated with TAC. It's basically suppressed at the level of sham with the use of GQ1. And when we withdraw GQ1, this gene is strongly upregulated again. So the next question was to perturb MIOX1 and see whether that affected fibroblast activation. We've done it in many ways in vitro with different experiments. I chose to show you one experiment in particular because I think it's very visual. We can culture cardiac fibroblasts in a dish. So we can take cardiac fibroblasts from a mouse heart, culture them in vitro, and in here you see fibroblasts, where in blue we are staining. So this is immunohistochemistry. We'll stain the nuclei. So the blue dots are the nuclei of the cells. And in green, we color this protein called alpha-SMA, that if you remember, I mentioned it as being a critical feature of the contractility of fibroblasts, which is hugely increased when these fibroblasts are activated. So this is the level of green that you have in an unstimulated fibroblast. Now, how do we stress the fibroblast in vitro? We use a signaling molecule called TGF-beta that mount a profibrotic response that mimic the fibroblast activation that's happening in vivo in the heart if we do, for example, TAC, if we trigger heart failure to a mouse. And you can appreciate how these cells are getting bigger and how they overexpress. They make much more alpha-SMA, and that's why the green signal, it's much higher. And that's what happens when we treat the cells with TGF-beta. So we stress them, but we suppress MIOX1. We just lower the level of MIOX1 protein. And you can see how this green signal, this alpha-SMA signal, it's strongly dampened when MIOX1 it's knocked down. And again, we've performed a series of in vitro assays, not only this one, to really prove that MIOX1 seems to be a critical activator of profibrotic function and fibroblast activation in vitro. Now, how is this relevant to human disease? MIOX1, I showed you, it's really beautifully regulated in the context of murine heart failure, but is it a regulator in human heart failure? It turns out that it is. It's very regulated. Here I'm showing you another single cell RNA-seq data from a collaborator. This data set, it's deposited in the preprint platform bioRxiv last year. And here you can see the fibroblast. It's the same concept. Every dot is a cell from donor patients, so no MI, and myocardial infarction, so patients with MI. And this is a density plot figure, meaning the darker is the color, the more cells are in a particular 2D space in this figure. So what this figure on the left means is that when you took fibroblasts from healthy individual, most of the fibroblasts cluster here in this lower part of the 2D space. But MI patients, the fibroblasts from MI, they instead cluster a lot in this upper part where you see this strong dark signal in this 2D space. And look at the expression of periostin and MIOX-1. First of all, the expression of MIOX-1 really seems to beautifully overlap the expression of periostin that, as I said before, it's really one of this classical marker of fibroblast activation. But you can also appreciate how most of the MIOX-1 expression comes from the cells of the MI patients rather than the donor, suggesting that, as it happens in mouse, MIOX-1 is poorly expressed in a healthy heart, and it's strongly activated in human heart failure. So to summarize what I told you about MIOX-1, what this work highlighted is how this critical regulator of pathological gene activation, BRD4, controls the activation of this novel gene MIOX-1. And we've done a lot of experiments to prove that once MIOX-1 is activated, it promotes the activation of hundreds of genes that are driving tissue fibrosis. And this really, it's able to make this cell state transition happen from a quiescent healthy fibroblast to a disease-activated fibroblast. So now my lab has made new mouse models that now will allow us to go from an in vitro experiment to an in vivo experiment, which we can perturb MIOX-1 in vivo in a mouse model of heart failure and see whether we can block fibrosis and we can promote a better healing of the heart. So that's something we're currently working right now. But I want to use the last 10, 15 minutes of the talk to tell you about an unpublished work that we have now in revision that tries to answer a question that I think it's very fascinating. So I show you how these fibroblasts are so sensitive to react to stress signals, but how are these cells sensing the stress? What are the upstream mechanisms that enable the fibroblasts to get activated upon stress? And in the last years, we've done a lot of work to try to identify what are the immune population that are important in activated fibroblasts in this context. And to make a long story short, we found a specific subtype of monocyte-derived macrophages marked by the expression of a receptor called CX3CR1 that seems to be critical for driving fibroblast activation and MIOX-1 activation in heart failure. So from now on, I will refer as this population as CX3CR1 positive because they express this gene called CX3CR1. So what was the critical experiment that let us believe that indeed the CX3CR1 positive immune population was important in heart failure pathogenesis? It's the following. So I told you about GE1. GE1 is a great tool. It allows us to inhibit BRD4 in vivo in a reversible manner, but it comes with a drawbacks. It's not cell-specific, right? We inject it intraperitoneal, and in the mice, GE1 will go in every cell in the body, not just in the cells of the heart. So we can't really distinguish what is the role of cardiac cells, let alone specific cell type within the heart. And that's where mouse genetics can really help. We made a new mouse models, a transgenic mouse model, in which we delete the gene that makes the BRD4 protein only in the CX3CR1 positive immune cells, meaning that this mouse express BRD4 in every cell in its body, but the CX3CR1 positive immune cells. And now I'm going to show you an experiment in which we subject these mice to this model of mouse heart failure, TAC, and I'll show you three groups with this color code. Sham is always in green. The TAC control where BRD4 it's not deleted in the immune population, it's in red, while this population where BRD4 it's gone, specifically in the CX3CL1 population, it's in blue. And that's what happens here in the y-axis. I'm showing you the left ventricle ejection fraction. These are the three groups. You can see nothing happens at baseline. But when we measure EF for 30-day post-TAC surgery, you can see this classical drop of function from the green control group to the TAC group in red. And you can see how the blue group is associated to a significantly higher EF, suggesting that this specific genetic perturbation, getting rid of BRD4 only in this CX3CL1 immune population, it's associated with increased EF, associated with a protective effect in heart failure. And this has been very interesting for us. How does it work? So what we noticed by performing single-cell RNA-seq experiment in the context of this experiment was the following. Not only there were many genes that were modulated by the loss of BRD4 in the immune population, and that made a lot of sense because the genetic perturbation was in the immune population. We found a lot of genes that were changing in the fibroblast. And that's what happened in the fibroblast. So I show you already single-cell RNA-seq results. So I'll go faster here, but I'll remind you here, every dot is a cell. These are fibroblasts, and where they're colored in red, it means these fibroblasts are coming from a TAC heart. That's the control heart failure. When they're coming from the fibroblast in which BRD4 has been deleted in the immune cells, they are colored in blue. And you can see basically they're distributed pretty far apart in this 2D space, right? They're on the right, the heart failure control in red, and on the left in blue, the BRD4 deletion. And on the right, I'm showing you again the expression of periostin and MIOX-1 in the same fibroblast. And here you have a legend that tells you basically that where you see purple, you have more expression of these genes. Where you see gray, you have less expression. And you can appreciate how most of the cells that express high level of periostin and MIOX-1, which at this time in the talk, I hope I convince you that are really very good proxy for fibroblast activation, are on the right side of this plot, meaning they're coming from red cells, from the TAC control. And this suggests that the deletion of BRD4 in the immune population, it's associated with a strong decrease of profibrotic gene expression. As you can see, most of these blue cells don't really express a high level of periostin and MIOX-1. How does it work? This really started to build this hypothesis that if many changes were happening in fibroblast, where the perturbation was happening in the immune population, that probably suggested that this immune population marked by CX3CL1 was sending some kind of critical signaling for activating the fibroblast. And how would we go around finding which signal that was? Well, of course, we were looking for a gene that was expressed in this specific immune population, the CX3CL1 positive one. Given that we were interested in this cell-to-cell interaction mechanism, we were selecting genes that encoded for a secreted factor. A secreted factor is a protein that a cell make and can spit out in a way from their cells in the extracellular space so that it can interact with receptors in other cells and drive a cellular response. So we filter for genes that made secreted factors. We were, of course, interested in genes that were strongly upregulated in the context of heart failure and that were suppressed with these two perturbations that I showed you in the talk, the drug-like perturbation with JE1, as well as the genetic perturbation with BRD4 deletion. And lastly, of course, we were interested in signaling was receptor was expressed at the fibroblast level, given that we were interested in a response in the fibroblast. And when we run all these analyses, it became pretty evident that a very intriguing candidate was interleukin-1 beta, I1 beta that encodes for a pro-inflammatory cytokine that all of you have heard about, that has a very important role in inflammation, in driving inflammatory responses in many diseases. We've performed a lot of experiments to prove that I1 beta seems to be indeed a critical factor for activating the fibroblast and the downstream profibrotic gene programs, such as the expression of MEOX1 and periostin. But I want to show you only one slide. This is the last data slide I'm showing you, which is probably the most relevant experiment for you. So what we've done here is to use a neutralizing antibody, an antibody that can sequester I1 beta so that it neutralizes its function and it's not able to engage with the I1 beta receptor in all the different cells. And these antibodies that we use in our lab are very similar to the monoclonal antibody that have been explored in studies and clinical trials such as the CANTOS, which has explored the role of I1 beta in the context of atherosclerosis. So what do we do with this neutralizing antibody? We went back to this model of murine heart failure by TAC, and we performed a surgery to trigger heart failure. And then for 30 days, we treat either with a control antibody, an isotype control, which will be in the group in red, or an antibody that's anti-I1 beta, that it's able to neutralize the function of I1 beta, and that's going to be in blue. And what we did was to measure cardiac function. So again, in the y-axis, the LV ejection fraction, you can see the two groups, again, baseline, no changes. And when we measure EF at day 30 post-hoc, we can see a significantly increase of EF in the mice that have been treated with the anti-I1 beta antibody, suggesting that by neutralizing this specific signal, we're able to protect in the context of this murine model of heart failure. And for the sake of time, I'm not showing you other results that also proved that the treatment with the anti-I1 beta antibody was also decreasing tissue fibrosis by histological assessment, as well as the activation of genes like MEOX1 and periostin that I show you being activator of fibroblast activation. So I want to finish it here with a final slide that really summarizes maybe the concept that I try to tell you about in the second part of the talk, and this immense and interesting biology related on how cells talk to each other in heart failure. I show you an example of a signal from immune cells that seems to be important in activating the fibroblast in heart failure pathogenesis, but there's so much more biology to be discovered. And for example, signals that activated fibroblast and immune cells in heart failure might send to cardiomyocyte or other cell types like vascular cells. So one of the research program of my lab, it's really focused on trying to understand what are the molecular mechanisms that are driving this pathological cellular crosstalk in heart failure, with the aim of identifying signals that are important for how the different cells talk to each other in disease, with the aim, of course, of not only identifying novel biology, but also maybe spurring the interest for therapies that can target these signals. And this is such a great moment in science to address this question, because we finally have the tools to dive deeper in the molecular response of these cells, even in vivo. And I show you example of how we can couple BRD4 inhibitor that we can give and stop giving in vivo with JQ1. I show you example of how we can delete a gene like BRD4 in a cell type specific manner, as well as we can couple all this perturbation with single cell genomics that allow us to measure genes at a resolution that we didn't have until a few years ago. So I'll stop it here with the for sure last important slide, highlighting all the people at Gladstone and elsewhere that have been incredibly important for my scientific growth. I want to mention the individual in my lab, but we just turned our first anniversary. We started our lab in September 2022, so we are 1 year old, and we have already amazing and passionate scientists in our lab. I want to mention the people at Gladstone that helped me a lot, Deepak in particular, and Arun that I mentioned before during the talk. And then we have amazing collaborators everywhere in the world who are helping us a lot in addressing this scientific question. And I'm happy to take any questions now, and I thank you for your attention.
Great. Thank you again, Dr. Alexanian, for bringing all your passion for biology and for presenting so eloquently a very complicated topic for the clinical cardiology audience. And I know that there are some basic science researcher amongst us, but it was a very elegant story. I guess I see that the chat is starting to be a little bit more active, but I can kick us off with just a question that I had. The story behind MEOX1 was very compelling. Obviously, fibrosis impacts other parts of the body. I assume that MEOX1 plays a role in other organ fibrosis. Has your group started to look into that, or what is known from that perspective?
Yeah, that's such a great question, and indeed that's a natural thing to look at. So what we've done in this context, and that's data we published in 2021, is that we took human primary fibroblasts from lung, liver, and kidney, which of course are notably associated with fibrotic diseases, and we stressed these cells with TGF-beta, and what we saw was the increase of MEOX1 expression in this context, exactly like we see when we stress cardiac fibroblasts in vitro with TGF-beta. There's also some in vivo data from recent single-cell RNA-seq studies looking at lung and liver, suggesting that the MEOX1 it's upregulated in liver and lung fibrosis. We haven't, of course, explored the function of MEOX1 in vivo in that context. We just made the mice for perturbing MEOX1 in vivo, but of course, a clear direction of the MEOX1 program. It's first starting from the heart, perturbing MEOX1 in murine models of heart failure, but then move to models of lung fibrosis, which we can induce with bleomycin treatment, or a model of liver fibrosis, which we can induce with NASH and specific diets. So that's such a great question, and it indeed highlights how this maladaptive programs associated to fibrosis, they're definitely very conserved also across tissue in different disease.
Thank you. Dr. Walker has a question also. Shannon, I'm not sure if you want to, I think I understand your question, but perhaps it's easiest for you to state your question directly.
Sure, yeah. Hi, thanks so much for a wonderful talk. If you don't mind, could you go back to the graph where you have, I think on the x-axis, it's time, and on the y-axis, it's LVEF. It's at the beginning. I think it shows the effect of the JQ1 inhibitor on EF recovery. My question related to that is kind of twofold. One, I'm just wondering what your hypothesis is as to why the EF gets better. I guess I'm thinking that if it's just halting fibroblast activation, there won't be further laying down of extracellular matrix and those types of things. So I guess I'm wondering why it just doesn't plateau at 45%, why it actually gets better, what the mechanism is of how it repairs itself. And then my other question was there ever a time period in the days after TAC where you don't actually see an improvement? I think that would probably have clinical implications of like, is there a time of irreparable damage where this just doesn't work anymore?
Yeah, and these are great questions. And going back to this graph that shows this really striking effect of JQ1, maybe I'll start by highlighting that I absolutely don't think that such dramatic changes in EF could be associated only in changes in the fibroblast or the immune population. I have to be honest, we don't understand all the biology in here. We don't understand why there's such a rapid modulation of EF. There's certainly much more dynamic changes in fibrosis than we initially thought. Indeed, even with a 2 weeks window, we see a clear re-increase of tissue fibrosis. So of course, there's a component of that, but what we believe, and going back to the last slide that I showed you about pathological crosstalk, is that there has to be some signals that maybe fibroblasts that when get activated, or immune cells are sending to the myocytes that could explain such a dramatic changes in contractility function. The myocyte don't seem to have a lot of genes changes, but that's only the transcription, right? It doesn't mean that functionally are not different. They're of course very different, because the EF looks very different. And for your last question, what we did was a crazy experiment in which, believe it or not, we had some TAC animal that we performed TAC and we haven't touched. We kept them in our mouse colony. And for some reason, some of them survived for a year. They had a very low ejection fraction, around their 25%. And we did this experiment in which we said, okay, let's take these mice and treat them for 15 days with AG1 and let's measure function. And what we've seen was there was a dramatic increase in EF in that context, going from around 25 to 45. So nowhere near the 60 that we achieved in the context of this experiment, suggesting that this increased EF driven by the inhibition of BRD4 seems to be age specific, given that it seemed to be happening also after a year. So I don't know if that answer at least in part your question.
Yeah, that's pretty cool. Thanks a lot.
Miki, that was a great talk. This is Javed running around in clinic. But I have a quick question about, do you think there is evolutionary an advantage for having MEOX1 around? What is the normal function of MEOX1, I guess? If obviously everything with fibrosis and the periosteum formation is bad, but what do you think evolutionary, why would we've developed, or
Yeah, that's a great question. And so let me start by saying something that I kind of left out of this presentation, that MEOX1 has an important role in development, early development. It's extremely important for somatogenesis and the polarization of the sclerotum. So a mouse model in which MEOX1 it's deleted all body will have craniofacial defects in ribs formation, and structure. So definitely MEOX1, it's very important in development, but that's another example of the many genes that have been studied in the last decades in cardiovascular science, of genes that are expressed in development, turn off in normal adult homeostasis, and seem to be reappearing in expression in disease. And of course, there's example of transcription factor that have been studied at CVRI and Gladstone for decades. So evolutionary, definitely there's a role of MEOX1 in development. What's the role of MEOX1 in disease and fibroblast response? Well, of course, fibroblast activation, it's a very conserved evolutionary mechanism that it's adaptive and allow us to repair when healing, right? If you get an injury in your skin, you need fibroblasts to get activated and make a scar, as well as you have an MI, you need some level of fibroblast activation and scar formation for repairing and prevent maybe the rupture. But what the fibroblasts are very bad in doing and understanding that this acute signal is finished and they have to stop being activated. And that's the problem with chronic stress, right? So there's no way for them to understand that these profibrotic genes like MEOX1 and periosteum, they have to be now turned on. And I guess that's a very intriguing evolutionary problem that we're trying to think about even in these terms.
I think that was a great answer. Thank you for the thoughtful question. Anyone, if we perhaps have time for one more question, else, I think we may just call it since we're nearing the hour.